Hey, this is the video for Biotechnology.2. So go ahead and set up a new entry in your Cornell notes. Remember to start a new page. And check out the essential vocabulary. Today we will be discussing genetic engineering, restriction enzyme, and gene splicing. So just to review, remember that a gene is a segment of DNA that codes for a particular protein. Remember that DNA is the instructions to make a protein. And a gene is literally just that part of the DNA that makes one particular protein. In this example, the protein that we're going to make is the protein for gray feathers in a pigeon, for example. Now, this process is way more complicated than I am explaining it to you, but I'm explaining it in a way that I think will make a lot more sense. So genetic engineering is a process that uses technology to change DNA by inserting genes. In this example, we will use, for example, I don't want a boring gray colored bird. Maybe I want something that's a little bit more extravagant, like the beautiful red colors that parrots have. I might want to take that gene for the red feathers and I might want to put it into the pigeon. Now what we will do is, this is a step-by-step process, and the first thing we'll do is we will use a restriction enzyme. And remember that enzymes are biological scissors and glue, and a restriction enzyme is an enzyme that cuts the DNA in a specific place for gene splicing. So in this example, we will cut out the gene for the gray feathers, and we will get rid of it. And then we will take the gene for the red feathers from the parrot and we will transfer it using chemicals. We will transfer it over into the parrot. So now this parrot has the DNA to code for making red feather proteins inserted into its DNA. We will use different restriction enzymes in order to glue that back into place. Now this is the process of gene splicing. And gene splicing is simply a process that combines DNA from two different organisms. When you splice something, you take it from one place and you put it somewhere else. So adding this DNA is not automatically going to change the bird. It's not going to change the colors of the parent. But if I were to modify this bird's sperm or eggs, what's going to happen? It's not going to change the bird itself, but when this bird has babies, guess what color the babies are going to be? When these eggs hatch, if the parent has this DNA, the offspring will have very similar DNA, and it will have that red colored gene. So this is a very simplified process for genetic engineering. And to review, here are the steps. The first thing is you're going to find a favorable gene to transfer. Then you're going to use restriction enzymes in order to remove that gene. You're going to insert that gene into a DNA segment through the process of gene splicing. And then you're going to allow that DNA to create the protein. Remember, DNA is the directions to make a protein. So what I would like you to do is respond to this check for understanding. I hope that this video was informative. I thank you for your attention, and I will see you soon.